He came with no preconceived notions about what chronic fatigue syndrome was. He also came with no preconceived notions about how the biology system works. He's like, okay, let's let the math do the work for us. And then it turns out that he's an amazing guy with math. So he's got an engineering background and a chemistry background that he brings to the biological sciences. And he's been just the most valuable member of our team since he's joined in and threw in with us. So I'm going to introduce Gordon Broderick from the University of Alberta, and he's going to tell you some of the work that's going on and some of the results that we have. Thank you.
Thank you very much, everyone. Thank you for your very, very kind hospitality. These weeks are always very exciting for me. They're very intense. I have to apologize if I stumble through this talk a little bit. We have been working very hard, as we always do when I come down here. And I very much appreciate your time, and the effort that you've made to come down and sit with us today. It's always very helpful and inspiring and educational for me to have these discussions with you and to meet with you. So what I've tried to do here, because we are very busy on several fronts, is to give you a bird's eye view of some of the things that we are doing. So what I've attempted to do here is to give an overview of the things that we are doing, and in doing so I may have sacrificed some of the depth. So I'm not trying to gloss over things. If you do have questions, please don't hesitate to slow me down. I'm also aware of time, and so I am trying to move through these slides at a reasonable pace, but please don't hesitate to stop me and ask questions. This meeting is for you. Pointing at the wrong, yeah, thank you, Nancy. An engineer that's technically challenged, that's a scary, scary thought. But one of the first things that I noticed when I became involved with CFS research were the sort of scatter of evidence that was being collected. Some of it was on the nervous system, some there were cognitive studies, there were sleep studies, then there were stress response studies with the HPA axis, so endocrine studies, and then was a lot of discussion about immune function, and conventional approaches to this would be, well, make up your mind. Is it a sleep disorder? Is it an endocrine disorder? Is it a cognitive disorder? Is it an immune disorder? And this is a relatively new field of medicine where we're now beginning to understand that it's not so cut and dry. Right? These are the three major control systems in the body. Right? The command and control, brain, central nervous system, the immune system, right, which also acts in very intelligent ways, and the endocrine system, which is a really broad control system for the body, controls everything from body temperature to appetite, et cetera, et cetera. And the more we study these systems, the more we now begin to understand that they're linked at a very basic biochemical level, that we have receptors on nerve cells for immune signaling proteins, and that, well, guess what? Immune cells can produce neuropeptides, right? And so there is this conversation between the three, and they do work in collaboration, in lockstep with one another, and they do have to maintain a certain balance, right? So this started us thinking about these kinds of complex chronic disorders as maybe a shift in balance of that exists between these three systems, right? A change in the conversation. Right? They're not having the same conversation between one another anymore. And I keep wanting to do this off my computer, but that would be foolish of me. There you go. And I think one of the areas where this shift in conversation is very, very visible is at the level of the immune system. Right? And the immune system is essentially like a social network. You can think of these diagrams, and again, I apologize, this talk was put together rather quickly. Right? So I'm not expecting you to read through these slides. I'd much rather we look at the pictures and we talk about what they mean. Right? You can look at those pictures and think of them as being the Facebook of the immune system. Right? You guys are all on. I'm not on Facebook. I'm a throwback to it. But you guys are modern and you're proactive and you're all on Facebook, and I can go look you up on Facebook, and then you've got your friends, and I can connect with your friends on Facebook. And the immune system is essentially composed of individual cells, right, of different types, right, and they speak to one another, and they crosstalk between the different types or different groups of cells, and then they talk to the broader system, the endocrine system and the central nervous system. Now, we talked about a shifting conversation. Right? What we did here is we looked at how we might be able to listen in on that conversation in chronic fatigue and compare it to the conversation that normally happens within the immune system of a healthy person. Right? Now, the graph here on the left is the Facebook or social network that you would expect in the immune system of a normal person and a healthy control. And the network, the social network that you have on the right is the network that you see in chronic fatigue individuals. Now, we've used some fancy pants math to identify those associations. Right? The thicker those lines are, the closer the association, the more information is being exchanged. Right? And every dot on that graph is an immune signaling protein. Right? So is a messenger. Right? So I mean, even looking at this just visually, just looking at it visually, you say, "Whoa, this is not the same communication network. It's not conducting the same business. It's not set up in the same way." Right? And that would be a pretty reasonable interpretation. Right? We can put some really fancy mathematical terms to it, but the bottom line is that's essentially true. That's what we're finding, and I can show you statistically that they are very, very different in terms of their architectures. Right? So it's a fingerprint. So if you listen to the immune conversation of a healthy person, you hear about, I don't know, last week's dog show, and if you listen to the immune conversation of a CFS person, you hear something completely different. Okay, and for those of you that are interested in the details of the immunology, essentially what we're seeing in the upper right-hand corner is the emergence of this Th2, right? This allergic response, which is suppressing or telling the antiviral response, well, don't worry, it's allergic. Relax, don't get all excited. So downplaying the antiviral response, the Th1 response, and we're seeing the NK cells, which are ever so important, which are part of the innate system as well, right, that are being disconnected from one of their early warning signals. Right? So less responsive to the early warning signals. So you're basically telling the NK cells, again, relax, don't worry about it, don't go tell your friends. Right? Which really, if you're a virus, is a great thing to do. Right? It's a wolf in sheep's clothing, and that's a brilliant thing to do if you're a virus, is to trick the immune system into thinking that you're just an allergen. Right? So what we're seeing here is kind of, it sort of makes sense from an evolutionary perspective. If you're a virus, I don't like viruses, but if you were a virus, that's kind of an interesting way to crash the party. Right? And so we're seeing evidence here of sort of a viral trickery in CFS, and that's manifested as somebody trying to seed the conversation and lead it in a different direction in the immune system. Oh, yes, go ahead.
Yeah, can you explain what you mean by link strength and what do you actually measure?
Sure. As I mentioned, every node in that network is a cytokine. Okay, so an immune protein. The immune system uses these proteins to signal to one another. Right? And what we're looking at here, that every link between those dots is essentially like a correlation. Okay? What we used is a very sensitive measure of association called mutual information. Right? So this was published actually in October, I think, of last year in Brain Behavior and Immunity. And some people have actually sent me emails asking questions about this. So it's out there. But essentially what we used is an information theoretical measure of information exchange or shared trends between those two cytokines. So if they move together in the same direction, right, and share a lot of the same trends, same or opposite directions, but as long as they do it consistently, dance consistently with one another, that line will be very thick between the two. Right? So you can see in the top cluster, very, very thick lines. Right? So a lot of coordinated activity. Thinner lines on the left-hand side. Why? Because, well, I'm just not responding to anything immunological. Right? So I haven't sounded the alarm. These people are under a state of chronic alarm. Right?
Basically, if you're looking at the link strength between two nodes, you're looking to see if they move up or down together.
That's right. That's right. As long as they do it.
In terms of their levels that you're measuring.
Exactly. As long as they change their levels consistently, I consider them to be dancing together. Right? Somebody's got to lead and somebody's got to follow, and sometimes they switch places, but they're doing it consistently. Okay? So we walk away with this saying, well, gee, that's sort of looks like a strategy that a virus would want to use. But we didn't have any molecular proof of viral infection in that cohort. Right? All we knew is that most of them were acute onset. Right? We didn't have a viral titers. We didn't have a firm diagnosis of an infection as a trigger. Right? So we took that information and moved on to another data set. This one is from a study that I'm principal investigator on that's funded by you. It's funded by the CFIDS Association. The following set of slides would not be possible to present. That research would not have been done had it not been for donor money, for donor contributions, and your support and encouragement. Okay? So what we've done here is pulled out of the freezer an NIH study where the samples had been stored, and this time what the investigators had done in that original study is brought people into this 2-year longitudinal assessment, but the inclusion criteria is that you were diagnosed with infectious mononucleosis. So we knew the trigger. Now we know that everybody in this study had EBV at the beginning of EBV infection at the beginning of the study, and then we tracked them through the 2 years. Right? And our control subjects are those that got better. Right? So we're comparing to those that got better. And at the end of 2 years, there were still about 5% of these are adolescents, about 5% of them were still fit the case definition for chronic fatigue syndrome after 2 years, and that floored me. That's 1 in 20. I have a 2-year-old daughter, and all of them happen to be young women. Right? So I have a 2-year-old daughter, and that scares me. Right? 1 in 20 is more than one person in a standard high school class. That after 2 years was still fit the case definition for chronic fatigue syndrome. Right? So we took a close look at these individuals, and we wanted to see if they all followed the same fatigue track, because we had documented their fatigue severity over 2 years. Right? And what we found is we were actually able to group them into 3 very distinct progressions of illness, okay, or trajectories of illness, to use the engineering term. And we found 3 groups. Right? The red. So what we have here on the right-hand side, is childhood fatigue as a function of time. These numbers are in months, from 0 months to 24 months. Okay, and the red dotted line are the recovering controls. Right? So they had infectious mononucleosis at entry into the study and were fatigued, but they got better. Right? So the red line is the individuals that got better. The other 3 lines are those that still fit the case definition for chronic fatigue syndrome at 24 months. And you can see that those 3 groups are quite different in how they changed in terms of fatigue level across those 24 months. The first group seems to be getting progressively better, but at 12 months starts to lose that momentum and almost finishes with an offset with respect to the controls. Right? The second group gets worse, but then has a nice downward trajectory to it. So they might actually, if we wait long enough, be getting better. Right? The first group, and I'm doing this in reverse order, I apologize. The first group, the blue line, gets better, but then gets a lot worse and keeps getting worse at 24 months. Okay. So we thought that was first of all, we were able to look at this only from the perspective of progression of severity of the illness. So now we want to know, well, we talked about biomarkers. Is there any way that we can measure this at the molecular level to see which track you might be on? That would be valuable, right? If I could do this at 12 months or at 6 months after your diagnosis with infectious mononucleosis, then I could pay special attention to you if I think you might be in a higher risk group. Right? So what we found, and the way we studied this again was to look at these cytokines, to look at these immune messenger proteins. Right? And what we found were that essentially only 3 out of the 16 that we tested for were different in their expression level at 24 months. Right? Only 3. However, these 3 enabled us very clearly to distinguish between the patient and the recovered controls. But it didn't allow us to distinguish which track you might be on. In order to do that, it was necessary to include 2 more immune messenger proteins, but these guys were not different between the controls in terms of level, of absolute level, were not different between the controls and the patients. So, following conventional wisdom, you would have thrown them out. You wouldn't have considered them anymore. Well, if they have the same interferon gamma level, that's not different. Let's move on. Well, again, the immune system is a social system, right? If you tell me how you're feeling, I have to compare it to how other people are feeling. Right? We have to put it in context, I guess is what I'm struggling to say here. The immune system is very context specific. Right? My IL-6 may be elevated, but may not be elevated. That might not be an elevated level for me because my IL-10 is also more elevated than normal. So the balance might be perfect for me. I just operate with the engine a little hotter than you do. Right? So comparing individual biomarkers and their strict level, you're going to miss some things that might be important. Yes?
Who are the C3s again that are a little more similar?
The C3s are the ones that have a decreasing trend for fatigue at 24 months, and we think they might be getting better. If we had followed them long enough, we think that they might be getting better. And we're doing some work looking back at other cases that we knew got better to make that comparison. And that's still very early to present the data. In fact, a lot of this stuff has only been presented once in Chicago, and a lot of the rest of the presentation has not been presented at all. So this is literally hot off the press. I was trying to give you the best and latest of our results and our thinking. So the C1s are essentially, that's my fault. The C1s are the ones that are getting worse. Right? The C2s are the ones that seem to be recovering early but then sort of plateau off and stop in their recovery. Right? The C3s are the ones that got worse at 12 months but then started recovering. Okay? And when we add, so that the IL-5, IL-8, and IL-23 are the ones that most people would look at because they were different in their levels between the controls and the patients. The interferon gamma and the IL-6 would not have been looked at because they were not different between the patients and the recovering controls, because the controls that we used in this case were individuals that had been diagnosed with infectious mononucleosis and were recovering normally. So they still had immune activation. They were recovering normally. Right? So that's why the difference is much more subtle here. Right? And when we add those two, we're not only able to distinguish between the controls, right? So this is a predicted severity or predicted membership of severity to post-infectious fatigue. We're not only able to separate the controls here, which have a low average probability of being sick, from the post-infectious fatigue as a group, but we're also able to tease out the first clinical course from the second clinical course, the C2s, I think this may have died on me, and the C3s that we think may be getting better seem to have a lower membership to the post-infectious fatigue group. So that, again, that seems to be consistent with what we saw in the clinical course. Okay. So essentially, what have we seen with these immune signaling proteins? Just to summarize, what we found is that in both the first data set I showed you, the first network I showed you, were with people that on average had been ill for about 10 years. Right? The second one, what I show you, the post-infectious fatigue, these are adolescents that had been ill for 2 years. Right? And so what we're seeing is that in both established and new onset chronic fatigue, there is measurable and very characteristic immune disturbance, and it's measurable. Okay. But it may not be measurable or easily usable if we only are chasing after one biomarker. Okay. It's a conversation, and I have to put things in context to be able to distinguish recovering people from those that are not recovering. Okay. So if I'm thinking, well, there's a different conversation between cells, right? Because this is what I was doing. I was listening to immune messenger proteins. Immune messenger proteins are being used between cells as a, could you pass this ticket to the girl at the back because I like her and I want to, and the ticket sort of goes through the class, right? Those are what the cytokines are, essentially. But what's driving that? What are driving those conversations? So we wanted to go a step further and see what might be underneath that. So we peeled into the individual immune cells, right? Sampled the blood, opened the zipper, pulled out the genetic material, right? Tossed it onto a microarray, and then measured, 25,000, the expression of 25,000 genes, right, in the immune system, right? So these genes will essentially program my cell behavior to produce certain signals and conduct business in the social network, right? So this supports, I said the immune conversation is shifted. Well, what are the processes in the individual cells that are supporting this shifted immune conversation? And so this is the technology that we used. Most of you are familiar with the word genomic technology or genome. This is what we're using here. So it's a fantastic technology. It's quite reproducible. Okay. But what we wanted to do also is depart away from looking at individual genes. The body has an amazing robustness to it. It's very adaptable, right? It conducts a number of functions, of metabolic functions, of signaling functions, of cognitive functions, right? But these functions are supported by more than just one gene. It's not a one gene, one function association. If it were, none of us would be standing here today. It would only take one mutation and we'd keel over, right? So it's a little bit like the space shuttle. The space shuttle goes up into orbit with 4 computers, right? Why not just one? One is fully capable of conducting all of the shuttle's computational needs. Well, the problem is, what if that computer goes on the fritz? Then you're not going to come home, right? So they use 4. And those 4 computers have to agree with one another, and they take a vote of majority to conduct that same function. Right? So that you can think of the gene network a little bit like the space shuttle, right? It's a really incredible design, right? You want to build those fail-safes in so that you can still continue walking around. If you go out into the sun and you get some sort of random mutation to an important gene, well, there are other genes and other pathways that will help you continue to conduct business without keeling over, right? And that's a kind of a nice thing to have. So they essentially work together in a coordinated fashion, and they conduct computation. You can think of the gene network essentially as a computer, right? And we know a little bit about the kinds of computations that the calculations that they do. Biochemists have spent an amazing amount of time, they spent entire careers characterizing these pathways, right? The way they conduct computation is to create these biological chemical molecules, right, that then get transformed through a series of enzyme reactions. And why do they use enzymes? Well, because we can't heat these things up. The body operates at a low temperature. You have to be able to conduct these reactions fairly efficiently. Enzymes help you do that. They're catalysts, right? So the genes support the production of these enzymes and conduct these very specific chemical reactions, and we know some of them. We've started cataloging some of these chemical reactions, right? So what we looked for here, rather than create a list of genes, which we know is inconsistent, and we know is susceptible to a lot of noise. Why? Because the body works with this high level of redundancy, right? So you shouldn't be looking at individual genes. You should at the very least be looking at groups of genes. And if we want to look at groups of genes, well, we should look at how they execute these known chemical reactions. And this is what we've done here, is that, for example, at this strange-looking diagram here, up at the top, are three genes, right, that the biochemists have told us, well, we know, we've validated this, that if this is up, this should be up. Right? If there's more of this and more of this, I should get more of this. Right? So we can now look through the data and see how well represented that known and chemically validated rule is in the data. Right? And so this is what we've done. And we've used, this was developed at the National Cancer Institute, and they call it the NCI Nature Pathway Interaction Database. Right? And this database contains a curated and validated by the NCI list of very basic chemical interactions that are supported by the gene regulatory network that we know to be true, or that we're fairly confident are true. Right? It's not an exhaustive list of all the interactions. It may be just the tip of the iceberg, but at least we know that they're true. So let's look for these truisms and see how well represented they are in the different patient groups. I mentioned that genes support different types of physiological activity, right? And they do that by activating these different rules. Right? So what I've shown here, I know then which rules support in general which activity. Right? And we're still looking here at the post-infectious fatigue individuals. Right? And when we look at the rules that are active or suppressed, that are more active or less active in these post-infectious fatigue patients at 24 months, this is the general split of function that they are supporting. Right? So big percentage of immune metabolism functions are different. Okay? Immune cytotoxic function, when I say immune function, I mean cytotoxic function, and the immune cell signaling, 28%. So those are the big slices of the pie. They just sort of jump right out at you. Right? Lack of vigor in the immune cells, lack of killing ability in the immune cells, right? And different immune signaling functions being conducted in the immune cells. Right? So this is consistent with what we saw with the immune messenger signals. Right? And we can impose all sorts of very strict statistical rules on this, and because the pathways pool the genes together, we can get very high levels of certainty. So when we looked then at individual pathways and said, well, that's really cool, which one of these 38 pathways that are differentially expressed between the post-infectious fatigue group and the control group, which ones are the most significant? And we can really tighten our noose and come down to 5 of these pathways. Right? 5 pathways are different in their level of activity between the post-infectious fatigue and the recovering controls, people that are recovering normally from infectious mononucleosis, and we can say that with a very high degree of consistency and reproducibility within this group of patients. Okay, so again, within this group of patients. What we need is a larger group, and we're going to continue that study with a larger group to be even more certain, but the statistics are very strong on even this group size. And what we're seeing are, three pathways out of the 5 that are increased. So, starch and sucrose metabolism, glycolysis, pentose phosphate, which is sort of upstream of phenylalanine, and 2 that are downregulated. Phenylalanine metabolism, which is responsible for tyrosine production, and I'll go to that in a minute, essentially is upstream of stress hormones. Okay. Interesting enough, upstream of stress hormones. Boy, go figure. Again, you've got tyrosine kinase, which is also linked to phenylalanine, which is also decreased. So it's kind of interesting that those 5 would be related. Right? They're all sitting around energy metabolism and signaling and stress hormones. Right? So which one, if I had to pick one, if you pinned me into a corner and said, well, that's really cool, Broderick, that's fine, but I don't like 5, I want 1. You're going to give me 1. You're going to give me 1 answer. Doctors like 1 answer. I'm sorry, I didn't mean that. I meant that in the friendliest of ways. I'm a bad man. If you forced me into a corner and said, well, okay, which one of these pathways is has the strongest statistics, or is maybe gives you the strongest signal to distinguish between the patients and the recovering controls, Well, the one that had the strongest statistics, again, in this data set, right, is the phenylalanine pathway. And that one is significantly depressed in the post-infectious fatigue individuals at 24 months compared to people who recover normally from infectious mononucleosis, right? The interesting thing here is not only is it significantly depressed in the group as a whole, but it's more depressed in the worst cases and less depressed in the C3 cluster that we think may be getting better. Right? That's interesting, because now telling me that I'm sick, I know that I'm sick. you don't have to tell me that. Comparing sick to healthy is an easy kill, right? Because in a lot of cases it's very, very obvious, and you see these massive differences in gene expression and pathways. Here we're comparing people who stay sick to people who were sick and are getting better. That's a more interesting problem, right? Because if I can get a bead on that, then I might be able to tell you, well, hang in there, or I'll pay special attention to you early in the game, because I know you're on a bad track, and I have to get you off that track onto another track that you will show that you'll eventually get better, right? So I'm looking for something that has some sort of predictive or prognostic value to it. Yes?
The phenylalanine metabolism has to do with the stress hormones or with what?
Yes, I'm sorry, I removed that slide in the interest of, I think I removed it. No, I didn't. I lie. I didn't remove it. It stayed in there. So it's the oops, sorry. I keep doing this, don't I? The phenylalanine metabolism is way up here. So upstream, you remember tyrosine kinase was another one, right? So they're sitting up here, right? And they lead down to L-DOPA, dopamine, norepinephrine, and epinephrine. Right? So they're sitting way upstream of epinephrine and norepinephrine. Okay. And that was suppressed in the PI-CFS. But I think what was most interesting to us was that the degree, the severity of suppression of that pathway aligned very nicely with the clinical course of fatigue. So as I mentioned, and I'll try and pick up the pace a little bit, the interesting part for us was that this didn't only distinguish sick from people who are continue to be sick from those who are recovering, but it also allowed us to maybe glimpse what track they might be on. And we continue to analyze this data. This is hot off the press. We have some early evidence that it might actually hold true, because we've gone back to the freezers and looked at some 12-month patients that we knew got better at 24, and then ran them through the same model, used the 12-month sample, ran it through the same model. And the results are encouraging, but we need to make very, very sure. So this is great, but this is post-infectious chronic fatigue, right? We're also doing a lot of work on Gulf War illness. And early on, it was made clear to me by Dr. Klimas that this is an important issue, because in terms of clinical presentation, there's a serious overlap in symptom. And the question now becomes, well, they're probably different illnesses, but how different are they at the molecular level, and how can we distinguish them more easily, right? And in order for me to get to know you, I can look at you, I get a first impression, we can sit there silently and sort of gauge one another, but I'm really going to get to know you if I start asking you questions. How are you? How old are you? What do you think? You can do that with the immune system too. You can challenge the immune system. That's the equivalent of opening a conversation and trying to, I shouldn't say interrogate, but ask questions and get to know how your immune system responds. And here we've used an exercise challenge to do exactly that, right? To see how your immune system, to probe your immune system, push it a little bit, and see how quickly it bounces back and in what direction. Right? So we've done that with the very, very valuable participation of the U.S. veterans that were deployed to the Gulf War. And we used a graded exercise test, and we measured immune cell populations, immune communication proteins. We even went down to the gene expression level. And so we did a very similar analysis to the one I just described for the post-infectious fatigue individuals. The first thing that we noticed, if we look at signaling networks, right, are again a very, very dramatic reorganization of the immune networks, even at rest. What was even more impressive was the way the immune network reorganized and responded to an exercise challenge, to effort, to additional effort. So let me just explain these boxes before we go further. These are, every dot here is either a cell population or an immune signaling protein. On the left-hand side, you've got the healthy controls. On the right-hand side, you've got the Gulf War patients. The bottom of the box is at rest before the exercise. The middle of the box is at peak effort. So we put them on these great veterans on a bicycle, and which is sounds cruel and is in some respects, but it's also very telling. Put them up to peak effort, then take a blood sample, then take them off the bike, let them rest for 4.5 hours, take another blood sample. Okay, so these 2 points are maybe 8 minutes apart. These 2 points are 4.5 hours apart. Right? And what we've done here is again look at how these cell populations and signaling molecules associate with one another and trend together and respond to one another. Right? And what we saw is a massive reorganization of the network to exercise challenge. The immune networks in the Gulf War patients were much more disorganized than the ones in the healthy controls. They needed to pull in all sorts of different resources. They were more resource hungry and much less efficient in their response to physical challenge than the healthy controls. Right? Then we wanted to know, well, is there any way that we can look across time and develop a means of identifying which one of the initial immune markers, has the biggest effect on exercise performance? And this is what we've shown here. And the one that came out top of the list, there are several. The one that came out top of the list is interleukin-1. Interleukin-1 has been studied and has been associated with sickness behavior, with fatigue, with problems in concentration. Keith Kelly is one of the, and Robert Dancer are two people who have studied this quite extensively. And it was interesting that we found exactly IL-1 in this population. And you can see on the left-hand side, IL-1 at rest, well, it trends with itself and it has a little bit of a splash effect on the rest of the network during the exercise challenge, but not really, right? And on the Gulf War side, IL-1 has a massive downstream effect on how the immune network reorganizes, and that effect keeps trickling through even post-effort. Even 4 hours later, you still see that connection, you still see that organization in the network. So again, we looked at the signaling, what supports that difference in signaling. So we went to the gene level, and we rather than look at individual genes, because there's so much connectivity between these genes, we wanted to see what functions, what pathways, what chemical reactions do they support. Also because that's what I treat when I treat you. Pharmacologically, I'm targeting an enzyme, a substrate. I'm shooting for a molecule that I'm either giving you more of or taking away from you. That's really what I do when I give you drug therapy. So it's important to work at the level of these chemical reactions because that's essentially what is druggable as an entry level. Okay? And what we found is when we compared the Gulf War patients with the asymptomatic veterans, that the biggest difference, the pathway that came out as the biggest discriminator between those two groups was a detoxification pathway. Okay, it was essentially a methyl naphthalene, which is naphthalene is a hydrocarbon. Right? My wife and I are chemical engineers, so she worked for a petroleum company as well. They usually put on quite a bit of protective gear to walk around refineries. So she was familiar with naphthalene. So that was interesting and surprising to us, and it actually, that pathway alone, only looking at how methylnaphthalene degradation pathway is active in circulating immune cells in these patients allows us to discriminate 80% of them from the healthy controls. And if we add to that two more pathways that are related to platelet activation and ATP processing, we get 90% accuracy in our assignment, in our discrimination of Gulf War versus the healthy controls. When we looked at the veterans with CFS, but that did not fall into the Gulf War category, we found, interestingly enough, phenylalanine. Right? So Gulf War veterans with CFS also have suppressed levels of phenylalanine, the same as the post-infectious mononucleosis patients did. So that was interesting to us, because now we've jumped genders and age groups. Right? So the real question now is, in these veterans with CFS and the veterans with Gulf War illness, is there anything that can be said about how different these two illnesses might be? Right? And when we compared those two groups, we came up with, again, very strong discrimination at the pathway level in pathways that have a lot to do with B-cell activity, with cell division, that have been associated with malignancies, and also pathways that support the synthesis and the repair of cartilage and connective tissue. And what we've done here is every single dot on these graphs is a pathway. Right? Pathways work together in chains. They hang out together as a group. Right? They're a really tight-knit posse. Right? The graph, every dot is a pathway, and the link, the edges between the graphs are the strength of association between those pathways. Right? In the upper right-hand corner, I seem to be having problems with the pointer. The upper right-hand corner, we have the control group. Then down at the bottom here, we've got the veterans with chronic fatigue syndrome, and then up on the left-hand side, we have the veterans with Gulf War. And again, even visually, you can see that they're not the same. Statistically, I can prove to you that they're not the same, right? And even by describing these networks in very, very coarse terms, like how connected are they? How many wires are there in these networks connecting the dots? Well, in CFS, I see that there are fewer wires connecting the dots than the healthy controls. And in Gulf War, there are more. Oh, that's kind of interesting. Could it be that I'm decoupling processes, that I'm cutting processes loose in CFS, and that I'm actually recruiting stuff, that I'm chronically overactive, and this may tie in with your comment on overactivity, chronically overactive in Gulf War. And this is what we did. So this might be a little bit easier to look at, I'm hoping at least. The vertical axis here, the midline of that graph, is essentially the healthy controls. Right? If I go to the right, I'm more connected, I'm better connected into the network, and every single line is a pathway. If I go to the left, I'm less connected with the greater network of pathways. Right? And isn't it interesting that CFS hangs out... Yeah, CFS is the blue, Gulf War is the red. So CFS goes to the left, is less connected in key pathways related to stress response, cell growth, cell repair, and immune signaling. Less connected, and the Gulf War, more connected in general. So more activity. Right? These people are sitting around a table and they're actively talking. They're talking more than the same pathways in CFS. And we can zoom in and look at these, and that's what we're in the process of doing, is now looking more closely at the architecture and identifying which of these pathways might be responsible, might be driving this reorganization of these metabolic and signaling pathways. And we're in the process of doing that. But essentially what we're seeing is the ones that are being reorganized the most or reassigned the most or increasing in their activity are associated with immune signaling, in particular B-cell signaling, repair and growth. Right? Inflammation's a good thing until you have too much of it, right? And signaling. And we see angiotensin there, which is interesting as well, right? Because we have autonomic issues in this patient population also. Why are we doing all this? Well, we're doing this because we want to develop treatments. Right? There are several things we want to do. We want to be able to identify people that are sick, right, as early as possible in the disease course, which is what I showed you at the beginning of the talk with the immune proteins. So with as few as 5 immune proteins, I might be able to give a fingerprint, and that fingerprint may have something to do with how your illness will develop. Wouldn't that be absolutely fabulous? Right? Still haven't cured you, but at least I've identified that you're at risk of getting worse. Right? And I've produced a molecular diagnosis at the molecular level. Okay? The second thing we did was we said, well, that we need to understand how. Before I can treat you, I need to identify entry points, things that need to be reset and rebalanced. So we looked at the network of pathways and we mapped these pathways out and we identified some candidates of things that are not in the proper balance with other pathways. Right? And what we're doing now is collecting this information and doing large-scale computer simulations where we're looking at tying up cortisol or giving you a bolus of ACTH in the hope that we will take your immune stress response system like an elastic band, right, pull it back and shoot you back into the proper operating mode, like a reset, right? Like just reset, you know, when your PC locks up, what do? You turn it off, you wait a minute, you turn it back on, and boom, hopefully it's back in the proper operating condition. And that's essentially what we're trying to do in a very conceptual way. And what I've shown here are some early diagrams of things that we are doing. We're looking specifically at the HPA axis, right? And our early models are focused on essentially giving ACTH or cortisol, tying it up or giving additional cortisol to see how we might be able to rebalance the HPA axis. And if we rebalance the HPA axis, well, something is going to happen with the immune system because the immune system sits at the bottom of that axis and feeds back. Right? So we're trying to reset. This slide here is focused on a hormonal reset, but we're also looking at resetting the immune system, which in turn might drag the HPA axis back into proper balance. So we're using computer simulations and quantitative models to do that. And these models require time course data. Right? The dynamics are important. For example, if I'm going to treat you, do I want to treat you in the morning or do I want to treat you in the evening? Right? If I'm a surfer, well, I want to ride that wave. If you have a natural wave to your system, I want to catch it and I want to push you because you're already moving in the direction that I want you to move in. Right? So these are questions that we're asking of ourselves, and so that's kind of where we stand now. And I just wanted to thank you first, because this work would not have been possible without patient donation. The preliminary data collected that enabled us to get these federal grants came from you, right? And by your donations, we were able to produce the science that attracted the attention of the NIH and the Department of Defense, and then roll out much larger dollars to enable us to carry it forward. So I want to thank you for doing that and for showing up this morning.